This week on the BackTable podcast.
So our device can actually be deployed in such a manner that it can cover the neck of some aneurysm types that would not typically be successfully treated by flow diversion. So it improves the chances that those aneurysms will go away over time. And the material itself is actually proving to be much less thrombogenic than metal as well, which is currently the main risk of placing a flow diverting stent into a blood vessel. So, there are other things like better imaging properties, allowing physicians to get more accurate information about an aneurysm in a parent vessel from non-invasive imaging like a CT or MRI scan after placement of the stent, as opposed to requiring an angiogram to get that same information.
Welcome to BackTable Innovation. You can find all our previous episodes on Spotify, Apple Podcasts, and at backtable.com. This is the next installment of BackTable Innovation, where we will learn from physicians and entrepreneurs working hard to drive healthcare forward. My name's Deanna, and I'll be your host this week. I'm a radiologist and biomedical engineer in London, and super excited to welcome Dr. Alim Mitha to talk more about biodegradable flow diverters and the future of care in interventional neurology. So welcome. So why don't we start? Why don't you introduce yourself to the audience? We can take it from there.
Great. Thank you. My name is Alim Mitha. I'm a cerebrovascular, endovascular, and skull-based neurosurgeon and biomedical engineer at the Foothills Medical Center in Calgary, Alberta, Canada. I'm also an associate professor of clinical neurosciences at the University of Calgary and faculty in the Department of Biomedical Engineering.
Why don't you tell me more about your journey? You've mentioned biomedical engineering, you've mentioned working at the University of Calgary. You gave us a very brief background, but I want to know more.
Yeah, I was born and raised in Calgary, Alberta, and I have a background in cell and molecular biology at the University of Michigan. I did medical school at the University of Alberta in Edmonton, Alberta, and the neurosurgical residency at the University of Calgary. Then I did a master's in biomedical engineering at Harvard University. I actually took some time out of my residency to complete that, and then following completion of my residency, I did 2 fellowships at the Barrow Neurological Institute in Phoenix. The first was cerebrovascular and skull-based surgery with Dr. Robert Spetzler, and then the second was endovascular neurosurgery, working with Cameron McDougall and Philippe Albuquerque. And then in Phoenix, I got married to my wife, Billy, who was doing a residency in pediatrics at the time, and then I joined here in Calgary as staff in 2011 and set up a research lab focused on tissue engineering and biomedical device development.
That sounds epic. So tell me more, what did that lab creation look like? You joined the staff, you said, in 2011. Did you go straight into having your own lab and working towards commercializing a product, or was that something that came later?
Yeah, so I always had a love for engineering and tinkering from a young age. And when I went to the University of Michigan, I actually lived on the engineering campus at the University of Michigan. And if you've ever been there, it's really grand but inviting at the same time.
Awesome. And when was the shift towards endovascular? Do you mainly work in endovascular, or do you still work sort of across the board?
Yeah, I work across the board. I still do open operations, a lot of skull-based tumors, pituitary tumors, and also do endovascular procedures.
Great. So interventional neuroradiology might be new to a lot of our listeners. So why don't we take it right from the top? Can you just briefly explain to us what is a flow diverter and what is its role in interventional neuroradiology?
Yeah, a flow diverter is a metal mesh that is placed inside the blood vessel to treat an aneurysm, as opposed to traditional ways of treating an aneurysm through the blood vessels, which includes coiling and requiring the catheter to get into the weakest part of the aneurysm. A flow diverter is very different in that it is placed in the main blood vessel, but not into the aneurysm itself. And it has enough material, typically metal, that allows it to divert blood flow away from the aneurysm, causing it to clot off.
Awesome. And is this essentially like an endoluminal reconstruction is kind of what's jumping into my mind?
Exactly. It's kind of a 2-phased approach to treating an aneurysm. The first phase is actually diverting the blood flow and causing the aneurysm to clot off. But then the second phase is that endoluminal reconstruction that you mentioned, which includes endothelial cells from the patient's body lining up over the aneurysm neck in order to completely exclude the aneurysm from the circulation.
Awesome. And how have they evolved? Is this a relatively new technology or something that's now quite well established?
Great question. So flow diverters started out as FDA approved in around 2011. They haven't evolved very much over the past decade or so. They have improved in terms of things like their delivery systems and in some of the materials that they are made with to make them more radio opaque or visible for the interventional neuroradiologist or neurosurgeon implanting the device. But overall, the structure of the stent itself has not evolved very much over the last decade.
And what about its indications? From the experience I have in medical device development, one of the biggest challenges that we have with new uses of something like a stent is making sure that it has that we can use it in the patients where we think they might benefit. So has this been the case for flow diverters? What kind of patients can you use these in? Any aneurysm, or is it quite selective?
Yeah. Flow diverters are typically used for patients with sidewall aneurysms or fusiform aneurysms. Initially, they were approved for aneurysms of the anterior circulation, so things like a superior hypophyseal aneurysm or a posterior communicating artery aneurysm or an ophthalmic aneurysm. But that indication is expanded to aneurysms in the posterior circulation and also fusiform aneurysms in the posterior circulation.
And can you just briefly explain what are the benefits of a flow diverter over, for example, a coiling or an alternative treatment option in this population?
Yeah, so the benefits of flow diversion are that you put the stent inside the main blood vessel and don't actually have to go into the weakest part of the aneurysm, which is the dome. Manipulating devices or putting coils into the dome of the aneurysm is probably the highest risk of the procedure, and a practitioner typically would like to avoid that if they can. There's a rupture rate of about 10% or so by putting devices into the aneurysm dome. Whereas a flow diverter, you stay in the main blood vessel, you simply deploy the flow diverter into the main blood vessel without putting a catheter into the dome, and it is much more safer from that perspective.
It makes a ton of sense.
Yeah, and the downside of flow diverters, of course, is that you're putting something into the main blood vessel, and something with quite a bit more metal than the blood vessel would be exposed to in the case of, say, aneurysm bridging stent. And so the thrombogenic risk is probably the biggest risk of a flow diverter.
And has this been the basis of sort of your research and the idea behind biodegradable flow diverters? Maybe we should just jump straight in. Why don't you just describe what it is you're building and what are the key advantages of a biodegradable flow diverter?
So I guess we're getting at kind of where did the idea for a bioabsorbable stent come from?
Yeah, let's start there.
During my training, whenever we considered placing a stent, we always considered the age of the patient. And I found it very interesting that we would always consider if the age of the patient, because we would be less likely to choose to put a stent in patients who are younger. And of course, all intravascular stents require antiplatelet agents. And in the decision-making process for how to treat, for instance, like a wide-necked aneurysm in an older patient, stenting or stent-assisted coiling seems like a reasonable first choice. But when you look at a younger patient, we would always put stenting lower down on the list because, and often, prioritizing surgical clipping in a younger patient over leaving anything behind in their blood vessel for a long period of time. This was even more an issue when flow diverting stents came on the market around 2011, since these stents have so much more metal and they require even a longer duration of dual antiplatelet therapy compared to the aneurysm bridging stent.
Just to be explicit, your concern is, them thrombosing off and causing a major stroke.
Exactly. And, we all know patients who have been on dual antiplatelet therapy and then they, transition to single antiplatelet therapy after a flow diverting stent, say even, the 6 months later, which is, when a lot of physicians recommend to stop one antiplatelet and just continue on the other antiplatelet, like aspirin. And then they stop the aspirin at some point for whatever, a surgical procedure, and then they get a thromboembolic event. And so they have to stay on the aspirin for the rest of their lives. And so, when you want to subject a younger patient to the requirement of being on an antiplatelet for the rest of their lives, you always think twice about doing that.
So just to bring you back to, where the idea came from, you mentioned, you're doing your neurosurgical residency, you're working as a neurosurgeon, and it's bizarre. You're in these MDT discussions, multidisciplinary discussions, and you're choosing not to treat young patients. So what was the next inspiration? Like, what made you think, I got to do something about this?
Thinking about how to make the technology more available and effective for younger patients without necessarily having them have to kind of have the long-term repercussions of antiplatelet therapy. The most obvious thing is to actually create a stent that serves its purpose for the time it needs to serve its purpose, and then just dissolve over time, so the patient can then not worry about having to take the blood thinners. And so at that time, kind of newer materials were coming out. There were other fields that were incorporating absorbable components into their implants and devices, and so it was logical to try to make an absorbable stent. It would have been very difficult to try to get some of those stents up into the intracranial circulation through tortuous pathways. So then we shifted our thinking a little bit and went to a braided design. This was all with the help of very talented people and students along the way. We designed and prototyped a braided bioabsorbable stent.
That's awesome. And tell me a little bit more about how it works. So you have a young patient, they have an aneurysm. You're using this stent like flow diverter, like any other flow diverter, right? You're bridging the main vessel to block off that aneurysm. How long does it last? How long when you're consenting a patient, how long can they expect it to be there for?
Yeah, so different materials can actually be tuned to last a different amount of time. And so what we were thinking about when we chose a material was how long it typically takes for a stent for an aneurysm to go away. And we know that there's data out there that says aneurysms typically take about 1 year or more to actually occlude after you put in a flow diverting stent, and that occlusion rate actually increases over the first couple of years. So we wanted our device to stick around for at least that long, and then go away. So that's how we kind of approached choosing a material. These materials nowadays can, like as I mentioned, you can choose a host of different polymers that have different mechanical characteristics, and based on their processing and their molecular weight, they can dissolve at different time points.
It's so great that this is something that we can tune and that we can put into patients. So, sort of drawing from the preclinical lab experiments, was this something that had to evolve over years? It sounds like, especially when you're working in the biodegradable space, it must have been a long journey.
Yeah, it sure has, and there's been a lot of learning along the way. So the development and prototype piece is only one piece of starting a business and trying to commercialize a device. I was fortunate enough to be working with a partner. His name is Sean Wong, and while I brought the technical background, he brought the management background and actually did a master's in business administration at Wharton. And together we were able to make this kind of a viable commercial startup entity.
And what did you do? So, you've been working in the lab. It started 2011. When did you incorporate a company, and when did you go and look for funding?
Yeah, so we incorporated in 2012. It took us a while to come up with a workable prototype. As I mentioned, we would iterate and implant them, find out what the biological response was. And in many cases, we had to go back to the drawing board and make design changes. And then eventually we had our workable prototype around 2019. The big thing that happened with us is we became involved with this startup incubator out of the University of Calgary called Creative Destruction Lab. And that really kick-started our thinking in terms of how to now take this prototype and raise money and then hire people and make it a business.
And where are you at now? I hear you have a team together, and there's lots of people behind this, taking this to the next step.
Yeah, exactly. So we are still primarily a preclinical company, but we recently started our first international, first in human clinical trial for the stent. So So we are now in patients. The clinical trial is ongoing, but preliminary results have been encouraging.
Nice. You'll have to come to the brain conference to find them out. I've seen you on the program. Yeah. So we briefly touched upon it earlier. You mentioned, we've got this option for a biodegradable flow diverter, but I'd love to understand more. What are the advantages? Like, what, as a physician, why would I choose a biodegradable flow diverter?
Yeah. So, over and above just the fact that it will absorb over time, and in the case of a persisting aneurysm, you could potentially reaccess that aneurysm, and the obvious benefits of being able to, say, discontinue your antiplatelet agents over time. It turns out there are more benefits than just those. Mechanical properties of metal are very different than polymer, and how the device actually behaves, for instance, or how it conforms to the anatomy is actually more favorable for the stents made out of polymer. So our device can actually be deployed in such a manner that it can cover the neck of some aneurysm types that would not typically be successfully treated by flow diversion. So it improves the chances that those aneurysms will go away over time. And the material itself is actually proving to be much less thrombogenic than metal as well, which is currently the main risk of placing a flow diverting stent into a blood vessel. So, there are other things like better imaging properties, allowing, physicians to get more accurate information about an aneurysm in a parent vessel from non-invasive imaging like a CT or MRI scan after placement of the stent, as opposed to requiring an angiogram to get that same information.
Let's get into the imaging a little bit more, because I find that really interesting. So talk us through why is having a polymer more beneficial to CT and MRI imaging? You mentioned that it has advantages, but can you explain it a little bit more?
So the bioabsorbable stent also has benefits in terms of imaging because there's much less, or in some cases, no metal in the bioabsorbable stent. So you don't have that artifact that you typically see with metal stents showing up on a CT scan or an MRI scan. Typically, a full metal stent, in order to get really good information about any residual aneurysm or the parent vessel, whether there's things like in-stent stenosis or thrombus, you need to do a digital subtraction angiogram, which is, of course, a more invasive procedure for the patient.
Yeah. And not to mention, if you put the stent in a young person, over their lifetime, they may need brain imaging for any other reason. And this allows them there's nothing here that's going to stop them from having that, which is amazing. Great to talk through some of the benefits of the biodegradable flow diverter. So you've briefly mentioned, this first in human trial, and I'm always curious to learn more about what a founder's thoughts are on clinical adoption. So, there's tons of innovators. They have loads of great ideas. And then when you put it out in the market, people love to ask questions. I mean, physicians are the same all over the world, always questioning, but why? What's the early feedback you've received from physicians? And do you think that there's going to be like a big challenge to overcome in getting this out there to the world once it is regulated?
Early feedback is very promising in terms of its deliverability. To be, we wanted to make it the same as kind of any other metal flow diverting stent on the market that physicians are used to deploying. And to actually increase adoption, we really have to prove out the value propositions, which we are currently in the process of doing. So I think that message will be very important about the benefits of a polymeric bioabsorbable stent compared to the metal ones. That'll be very important for early adoption.
So we're not using metal, right? What materials can you use that are stiff enough and have an appropriate mesh density, length, deployability, such that they can function as a flow diverter?
I think newer materials will be developed over time that can serve the purpose even better than the materials we have available now. So currently the materials that we're looking at now to create bioabsorbable stents include things like poly-L-lactic acid, polyglycolic acid, polycaprolactone, things like that, which are polymers that essentially degrade over time.
And are they stiff enough to achieve what a flow diverter does?
Yeah, that's a great question. Of course, you don't want too much stiffness because metal can sometimes cause bad things to blood vessels if they're too stiff. Things like intimal hyperplasia and changing the direction of blood flow because of their stiffness, or causing the blood flow to adapt more to the stent's configuration as opposed to adapting more to the blood vessel. But in general, the polymers are a little bit softer and they are a little bit more yielding. So they're less likely to put as much radial force on the inside of a blood vessel, but more likely to conform to the blood vessel.
And I can imagine that's a huge benefit, especially in the small arteries that are in the brain.
Exactly.
And what about its sort of its deployability? Does using polymers create a problem with the delivery system, or is it quite compatible?
So using polymers in terms of delivering them is interesting because you don't actually find that you need as stiff of a delivery catheter to get the polymeric stent into place. So in general, today's catheters are over-engineered for bioabsorbable stents that may be coming out in the future.
Awesome. And sort of in the preclinical lab experiments or in the trials, in the experiments that you've been involved in, how long does it take to degrade? So if you put this into, say, a 35-year-old, when can they expect this to be resorbed?
These stents can be made out of materials that are actually chosen based on the length of time it takes for them to degrade. And of course, that in turn is typically chosen based on the application of the stent. So in the case of a flow-diverting stent for an aneurysm, you usually want the absorbable material to stick around for as long as it typically takes an aneurysm to heal, which is about a year or up to 2 years, in that range. So at that point, you want the stent to dissolve, or most of the stent to dissolve. If you choose a material that dissolves too quickly, then there can be issues with potentially aneurysms recurring or not being fully treated, meaning that the endothelial cells don't actually line up and create that new blood vessel wall or parent vessel wall that you want to fully exclude the aneurysm from the circulation. So I would think in this application, you would want the stent to stick around for at least a couple of years.
Is material science has it advanced enough so that we can be confident that it's only going to last 2 years, or is this something that, is still to be answered?
Material science has actually come a long way, and you can choose the same material and actually get a different molecular weight out of that material depending on how you process it. And then that molecular weight actually in turn feeds into how long it will take for it to absorb. So you can certainly choose materials and specifically their molecular weight to actually design when you want the stent to go away.
I'd love to hear more about the prototyping stage, my personal favorite part. It's all of ours, isn't it? You mentioned that you and John worked together to create something physical that you could show investors, and we can't underestimate how powerful that is. I know a lot of our listeners have their heads bustling with ideas. What was it that helped you build that prototype? Was it being affiliated to an academic institution? Was it the fact that you're doing these procedures on a day-to-day basis? Or did you have the inherent skill set from your biomedical engineering background?
You always leave your academic program having some skills, but never all the skills to actually do what you sometimes envision you want to do. So mine is in biomedical engineering, and I came together with John Wong, who is my partner at work, and he's also an endovascular neurosurgeon. And his skill set was in management, and so he actually did a business degree, an MBA, an executive MBA through Wharton, which was very impressive because he did it during while he was a staff person. But essentially, what we had to do to get to the prototype stage was essentially work through my laboratory. So I have a laboratory at the University of Calgary. It's focused on biomedical engineering and biomedical device development. We also have some tissue engineering projects going on in the lab, but we have numerous talented students, including PhDs and master's students who helped us get to this point along the way. Essentially, the idea morphed over time. Initially, we tried to create a laser cut stent, but polymers can actually be too brittle for doing that properly. They can be stiff when created using lasers, and then very difficult to get up through the tortuous anatomy. And so at some point we shifted to a braided type of flow diverting stent, and then essentially just kind of bootstrapped that mission to get a prototype completed. Failed a couple of times, and then finally got a working prototype.
And I'm sure that must have been a great moment where you could take this physical flow diverter to the accelerator that you mentioned. And we can't, like I said previously, you can't underestimate how powerful that is.
Absolutely. The interesting thing about it is that a stent is only a part of the final device. The delivery system was actually the most complicated part, even though it's not the part that you leave behind. So we had to do a lot of learning in terms of how to actually deliver what we had prototyped in terms of the stent.
And what were the main challenges? I'd love to know.
Yeah, creating a delivery system. Of course, polymers are very different in terms of how they behave, as you mentioned. Just like they're not as stiff as metal, and so you have to create a delivery system that kind of treats the polymer gently and is able to get it up to where you need it to go.
It sounds like you had to rationalize the engineering that had been done previously.
Exactly, yeah. There's always, you always kind of develop something, and the first thing you do is learn what's out there already. And so there was a lot of learning to do.
Nice. So you've mentioned already that, you've got your first in human trial. And after that, the big hurdle's always regulatory. So is the aim for this first in human trial to feed into that?
Yeah, so our first in human trial is just a few patients. We're trying to learn as much as we can about the device and how it behaves. Our goal is to get, just a few patients so that our primary outcome measures would be things like technical feasibility and safety. They're always the first kind of two things we want to learn the most about our device in terms of a first in human trial, and also get efficacy information as well. The goal after that is to do a larger kind of efficacy trial where we learn about, once we prove safety, just to learn about the efficacy of our device and compare it to some of the existing devices on the market.
And what do you think is going to be the biggest challenge in achieving that scale up? I know it's difficult to say because, you've only really got preclinical data today, but what do you think is going to be the biggest challenge?
The biggest challenge in terms of kind of taking it to the next clinical trial? Is that the question?
Yeah, I mean, I guess what I'm trying to hint at is, do you think clinical adoption might be a challenge here? So considering how this is such a novel approach with a novel delivery system, do you think you're going to have any hurdles to overcome when thinking about clinical adoption?
Yeah, that's a really good question too, because physicians have to have a different mindset and a different purpose for putting in a flow diverter that is absorbable versus one that is permanent. Of course, ease of use is paramount. Physicians have to be able to use this easily and not requiring any other kind of resources compared to the existing flow diverting stents. Has to be straightforward and using similar size catheters as everyone is used to, but they have to see real benefit in it. And that's what we're hoping to do with the clinical trials, is actually show that there's much more benefits to an absorbable stent compared to traditional metal ones.
And what's the early feedback you have had from physicians and potential future collaborators?
Yeah, early feedback has been very good. There are definitely some other advantages of an absorbable stent over and above what you would think of in terms of it just going away for the most part over time. Things like its mechanical properties, or being able to retreat an aneurysm if it does stick around after flow diversion, because that's one of the biggest value propositions also to an absorbable stent, is that you don't permanently lose access to the dome or the sac of the aneurysm. It actually, you can re-access it, whereas with a metal flow diverting stent, you've essentially lost complete access to the sac of the aneurysm in the case that it should stay persistent.
Great. And so what advice would you give to new docs or established docs that have an idea and they want to, take it from that 0 to 1 that we were mentioning? What would be your golden tips?
Okay, so there are a couple things in terms of, for others who want to start on, say, an entrepreneurial journey, they have a good idea and they want to implement it. I would say it's very difficult to do it on your own. Get a good partner. There are a lot of decision points along the way, and having a sounding board in someone who doesn't necessarily think the same way as you do, but has a common goal in mind is very helpful. So I think that would be one piece of advice. There will be ups and downs along the way, and try to enjoy the entire experience, because ultimately you should be doing this to try to give yourself some sort of satisfaction that even for a short period of time, you've actually made the world a better place or improved patient outcomes. So it's a challenging journey, managing people, learning new skills, but it is definitely worth it in the end, having tried it.
Yeah, definitely worth trying to get from that 0 to 1. And to your point, making sure that you find other partners with a skill set. And that's sort of what we touched upon earlier, right? It's the PhDs and the master students that are working in your lab. And you know what? They're not physicians. Like, their expertise is not the patient care and the clinical adoption. Their expertise is in overcoming those manufacturing challenges and really taking a step forward. That's what really takes things forward and allows and unlocks the funding that we also touched upon. I'd love to understand a little bit more about what the setup looks like with the University of Calgary. I've heard great things. I hear that it's a bed of innovation.
It actually is. I don't know if everyone working in an academic environment can actually say this, but the University of Calgary is one of the best places in North America right now to go from academic research to commercialization. It's interesting because in this geographic area in Canada, we've been, traditionally focused on oil and gas as the main driver of our economy, but partly in response to political and environmental policy changes that are shifting the way we think. Calgary, in particular, the University of Calgary is really leading the way in terms of trying to create opportunities and elevate other economic drivers in the local community. So like life sciences and technology. So University of Calgary is actually Canada's #1 startup creator in all of Canada. And this is mainly because it creates a favorable startup environment, things like removing red tape, facilitating IP protection, but allowing the creators of the IP to own their own work, creating centers for innovation within the university, including physical spaces, and even investing in the startups that they create. And many universities, on the other hand, mandate that the IP be filed and assigned to the university and then licensed by the creators. So we have a very favorable environment for that, and I'm really fortunate to be making this effort of commercializing our stent technology from where we are, and it's played a huge role in our success so far. I was focused on medicine, but I briefly considered transferring into engineering.
Me too.
But unfortunately, I wouldn't have been able to fit in all my pre-med courses, so I bailed on that idea, and like you, then decided to do bioengineering at a later stage. But I was actually fortunate to do that, because then I thought about bioengineering again after I started neurosurgical residency and realized that, especially neurointervention is really heavily focused on biomedical devices, whether it be the implants or the delivery systems, catheters, things like that. But more than that, I think I realized that these devices don't always work the way the physician wants them to, and more importantly, they don't always work the way the patient wants them to, meaning that they're not always effective. So during my residency, it became really apparent to me that there's a lot of room for improvement with some of these devices. And so that's when I kind of took a few years off of clinical residency, went back to do a master's degree in biomedical engineering at Harvard University, and worked with some great people, including Chris Ogilvie at the Mass General Hospital. He's a very good mentor and friend, and he really facilitated and fostered my interest in novel treatments. And then in his lab, and with the help of Brian Ho, is another dear friend, I learned the modified elastase model that Brian initially described. So when I started on staff here, I actually brought that model back to Calgary and set it up in my lab. And having that lab piece to the company has been really important. Once we developed this idea and kind of cultivated it to the form that it was into kind of an early prototype stage, we obviously had to learn about how it would behave in a biological system, and having the in vivo model at our disposal through the lab really helped us to iterate the device very quickly, which doesn't always happen in kind of an industry alone setting. So having that kind of that lab piece really helped us to refine the prototypes and do things like refine the delivery system and kind of advance the product to where it is.
That's great. And it's good that you've had promising early feedback. And it sounds like you have a pipeline of clinical trials that are going to lead to this adoption. It's very easy to just think about the first in human and not have the pipeline of evidence generation that's going to lead to this flow diverter being available in every single neuro-IR angio suite in the world. So very happy to hear more about that, and super excited to see the trials in action, because they'll be big ones.
Yes, it will be. It's really exciting. We're taking one small step at a time, but of course we have high hopes for this, and are looking forward to seeing how the rest of the story unfolds.
Me too. It's been great to have you on the show. Really incredible to learn more about biodegradable flow diverters, and it's been great to hear your insights of how you can turn a medical degree and a biomedical engineering degree into a product that is out there and will be making a difference to patients pending these exciting results trials. It's been super great to learn more about biodegradable flow diverters and the incredible career and onwards journey of where you guys are heading. So thank you for coming on BackTable, and really excited to learn more and meet you in person at Brain.
All right, well, thanks for having me, and I look forward to seeing you and everyone else at Brain in December.
Thank you so much for listening. If you haven't already, make sure to subscribe, rate the podcast 5 stars, and share with a friend. If you have any questions or comments, DM us at BackTable Innovation on Instagram, LinkedIn, or Twitter. BackTable Innovation is produced and hosted by Brian Hartley,
Aaron Fritz,
Diana Velazquez-Pimentel,
and Eric Amaker.
Our audio team is led by Kieran Gannon, with support from
Josh McWhirter,
Aaron Bowles, Nick Shellcross,
and Ness Smith-Savadoff. Design and digital marketing led by Brian Schmitz.
Social media and PR by Ann Dang.
Administrative support provided by Jamila Kennebrew.
Thanks again for listening.
See you again next week.